the living system around the human beings, the living systems around the animals' bodies, including plants and human life. Develop the mechanistic understanding of the effects and ensure the safer chemical products. The role of toxicologists is develop the safer drugs and medicines. Let's take a very common example of हरे पत्ते की गोली. You heard about नीले पत्ते की गोली, हरे पत्ते की गोली दे दो. अब आप थिंक करें, सोचें, केमिस्ट ने आपको हरे पत्ते की गोली दी. अब हरे पत्ते की गोली की हरे कलर की, ग्रीन कलर की स्ट्रिप, बहुत सारी टैबलेट्स, बहुत सारी स्ट्रिप्स आती हैं ग्रीन कलर की स्ट्रिप्स में. अगर केमिस्ट ने आपको कुछ पैरासीटामोल के बदले कुछ अलग मेडिसिन दी, इफ देन व्हाट विल हैपन? तो इट इज द ड्यूटी ऑफ द एंड यूजर्स आल्सो टू एंश्योर द सेफ्टी. हरे पत्ते की गोली, नीले पत्ते की गोली इज नॉट द नेम ऑफ द मेडिसिन. तो व्हाट वी से द पैरासीटामोल. पैरासीटामोल इज अ एंटीपायरेटिक OTC मेडिकेशन, ओवर द काउंटर. आप जाते हो and it is a safe for the use for the major population, that's why it is easily available. But the same drug, the paracetamols, can be toxic to you if given in a high dose, more than 4 gram in a day. It will cause hepatotoxicity in the body. The same drug that is used for the antipyretic actions for treating the fever, for lowering down your body temperatures. Now come to another example, that's tetracycline. Tetracycline is a class of antibiotic that is used for killing the bacteria in the body. You are suffering from several disorders, bacterial disorders, the doctors prescribe the tetracycline, oxytetracycline, tetracycline. Now after prescribing, doctors and the pharmacist had given you the tetracycline. Now he did not inform any guidelines, he did not advise how to take the tetracycline. Tetracycline have a very significant drug food interaction. Drug food interactions. Food can be milk, can be any electrolytes, and as the tetracycline interacting with the food materials, particularly the calcium part, binding with the calcium, tetracycline is binding with the calcium, and after reaching in the blood, it is causing yellowing in teeth in children. It means if any doctor prescribe the tetracycline, it will cause deformed teeth and yellowing in the teeth that will lead a toxicity in your kids. So it is mandatory for a doctor and for a pharmacist to make sure the safe administrations of a drug for end users. Let's take another example, is a Nimesulide. Nimesulide is a drug used for the antibiotic across the globe, but after thoroughly inspections, after thoroughly reinvestigations over the animals preclinical and the clinicals, it was found that it is causing hepatotoxicity in children and in adults. Thus, European nations and US FDA thus block the use of Nimesulide across the globe. Next, determine the risk from the chemical exposures. What do you mean by the determine the risk from the chemical exposures? Look around yourself. Everything around me is composed of some kind of chemicals. We are around. What I am wearing is made from some chemicals. Light is also another form of radiations. The table, chair, blackboard, everything is around is any kind of chemicals. So we cannot avoid any chemical exposure to us. But our duty is to ensure the safety of the ourselves from these chemicals, and prior to launch in the market, prior to use in the household, prior to use in a living system, we ensure the safety. Next is develop the treatments for chemical exposures. Next is ensure a safe food and water supply. Now, in the zone of social media, social sites, everybody used to Zomato, Swiggy, and other social sites for ordering the food. But my concern over the quality of the food, how they are making the food at their restaurants, at their cafes, we are not sure the hygiene and the water supply they are using. There is a prime challenge and the social concern over the quality of food and water used in these formulation, and particularly the oil. The oil is used in every product, either Chinese, Indian, non-Indian, Mughlai, any kind of food they used a large amount of oil, and the quality of oil is leading to cardiovascular disease across the globe, and our country become number one in lifestyle disease, particularly cardiovascular disease, and we are overburdened with this disease. Thus it becomes our moral responsibility to pay the attention towards the quality of foods so that we can avoid the economic burden on the economy. Next is forensics. How the role of toxicologists play important role in the forensics for crime scenes, for determinations of the criminals is used. We use forensic toxicologists. Thus, we have a different role of toxicologists. Thus, the toxicology is a melange of various disciplines, and every discipline is claiming that he make a toxicologist for the society, but it is not true. The toxicology comes from every domain of the science, humanities, from biostatisticians. We require the toxicology from the every domain. It's a melange of science. Toxicology is an applied science with many areas of specializations. We require the chemical, chemist, pharmacist, molecular toxicologist, pathologist, histopathologist, food experts, regulatory agencies, the instrumentations technologies and instrument analyst. We require every expert for a good outcome. Thus, it become an applied science with many areas of specializations. Who are toxicologists? The toxicology involves integration of information from many different areas of expertise. People working in toxicology can also be called the biochemist. They are working with the living cells. The chemist, pathologist, cancer researchers, veterinarians, the medical doctors, or the clinical toxicologists, cell and molecular biologists, engineers, mathematicians, statisticians, lab technicians, animal care providers. These are all toxicologists. We require hand to hand, one to one, and everybody's contributions in developing a drug, a molecule, or food products for the safety assurance for the safety of the food products. What are the major areas of the specialization in toxicologies? Broadly, we have three major parts. First is mechanistic toxicology. Others way we say the basic biology and the chemistry. Second is descriptive biology. Others name is testing toxicology. Last is regulatory toxicology. The regulatory toxicology is mean for making the rule and compliance for a new molecule for already existing molecule. What are the major areas of specializations in toxicology? The risk assessment, the developing of the modeling, the translationals and clinical, applying the basic research to patient care. Now come to the major point, the risk assessment modeling. The risk assessment modeling can be done in two different methods. First is preclinical studies, second is clinical studies. In preclinical studies, we have a different models for a different disease, and further we can classify the preclinical studies into three major parts. First is in silico studies, second is in vitro studies, third is in vivo studies. In silico studies, when we have a new molecule, new structures, we can draw the chemical in the laptop in the some software. With the help of computer, we interact that molecule with the live cells, with the receptors. So, we are using the computer, we are using our molecules inside the computers, and we are developing a receptors. We are interacting of molecule with the receptors within the computer. This is called the in silico studies. After interactions, there is yield some data that is termed as docking score, and docking score is used for the toxicological evaluations. If the score is less than 4, it is less potent. If the score is more than 6, it is semi potent. If the score more than 10, it becomes highly potent and can be used for further investigations. Next is a synthesis of the chemical. Now the chemist has a role in the synthesis of the chemical. After the chemical, we required analyst. Analyst check the purity of the chemical and others structures validations of the new synthetic molecules. After this, we have a in vitro study. The newly synthesized molecule is tested for in vitro studies. Now, what is in vitro study? In vitro study, normally we use live cells. Cells could be of cancer cells, could be normal liver cells, of any skin cells that can be used, and we check the interactions of drug, of new molecules, new entity, new chemical entity with these cells present in the petri dish in 96 well plate, and they are further studied for their molecule assays for molecular pathogenesis. Next is translationals. Next is in vivo study. If the molecule is passed from the in vitro study, next we plan the acute toxicity. There are the various types of toxicities based on the severity. First is acute toxicity, second is subacute toxicity, third is chronic toxicity. Acute toxicity based on the days. Within the you have taken any molecules for, and it has a side effect within the 24 hours. Immediately उसका side effect show हो गया है. Then the molecule suspect for the acute toxicity. और इसका इफेक्ट क्या है? बहुत जल्दी we can treat. जितनी जल्दी इसका इफेक्ट आता है, इतनी जल्दी इसको हम treat करते हैं. It means it is acute effect. If a molecule having a longer effect after 1 month, after 2 months, after 3 years, after 2 years, then becomes a chronic toxicity studies. And in between these two, we have a subacute toxicity. So we have a 3 grade of toxicity based on the severity. First is acute, second is subacute, third is chronic toxicity. These are developed for various models based on the molecule availability. Next is translationals and clinical. They are applying basic research to patient care. Now, come to the first point, the mechanistic toxicology. Focus on how the chemical produce adverse effects, how the chemical is imbibed in the body, how the chemical is interacting with the receptors, and which receptors. We have a numerous receptors in our body, and how the chemical is interacting with these molecules. Is there any chromosomal change? Is there any genetic change? Is there any mitochondrial change? Is any deformities occur within the cells? This is the main work of mechanistic toxicology, to find out, to investigate the role by biochemical interactions of a molecule with the receptors. The biological system protect themselves against the adverse effects. Now, it involves the cellular and the molecular biology. In cellular and molecular biology, we have lots of instrument available for studying these kind of the studies. Next, we have a xenobiotic chemistry upon the xenobiotic metabolism. Xenobiotic metabolism. What is xenobiotics? Xenobiotic is a chemical that is foreign to the organisms. Any molecule entering into our body termed as the xenobiotic. A drug, a paracetamol, a food, any chemical entity which is inhaling through inhaling or eating termed as the xenobiotic, and causing any side effect in the body. Now, mechanistic toxicology. Chemical research in toxicology usually investigate metabolic transformation of drugs or potentially hazardous chemicals. We have taken a chemical, let's say ciprofloxacin. Ciprofloxacin is a potent antibiotic. After taking into the body, it is metabolized in the various forms so that it can give their pharmacological effect in the body and kill the microbacteria in the gut. But how the chemical is reacting, how the antibiotic ciprofloxacin is interacting in the body, how persistent is the chemical in the body, are metabolic product toxic? In study, we found there is no toxicity of the ciprofloxacin, but it may be possible after entering in the body, after presence of some food, after presence of any drug, it may lead to any toxic product in the body. So, this is the role of mechanistic toxicology. We are finding the mechanism of toxicology at a molecular and cellular level. Now, do test animal exhibit the same result as the humans or the other species of the concerns? Initially, whenever we perform any toxicological study, we usually perform in animals, as the animals nearly symbolic to the humans and their 95% genetic makeup is to the human beings. No, the most commonly animals use are the rodents. Rodents have a different species mouse, rats, depending on the models. We can use Vista rat, we can use Pagtoli, we can use Swiss albino mouse, and for cancer studies we use nude mice, Balbisi, and different other species for the cancer studies. The first is identification of a right model for conducting the toxicological study. For example, we are studying the anti-vomiting activity. Anti-vomiting activity means any drug that can bring vomiting stop, or that can stop the vomiting in the humans. But this study cannot be studied in the mouse. Why? Because mouse has no CTZ center. CTZ center stand for chemotrigger zone. When the animal has no CTZ, so we cannot use this model for conducting the anti-emetic or anti-vomiting activity. First is validations of the animal model for conducting the toxicological study, and right model is essential for conducting the toxicological study. So, for the CTZ we use rabbits and other species like monkey and other species for conducting the anti-vomiting activity. Next is the descriptive toxicology. Typically involve the toxicity testing. Broad spectrum of responses reflect the toxicity. Functional effects such as immunological responses. Developing a different models for immunological response. First is triggering the immunological response. How a new molecule, how a new chemical entity is entering in the immunological response. For example, COVID vaccines. It, as a new molecule in the country, it can relate, it can initiate new immunological response. The first is to ensure the role of vaccines in the human body, particularly for the immunity, as it is reacting, it is affecting with the immune system of the human body. Next is growth inhibition, as the new molecules interfering with the growth inhibition of the human life or life. Reproductive impairment. Reproductive impairment, and a new molecule is affecting the sexual life of a human being, of animals, or bringing infertility or fertility in the humans. Increase in cancer incidence, mortality. Descriptive toxicology perform these kind of studies for the smooth launch, smooth marketing of a new molecule. Next is assess the concentration dependent hazard a chemical may present, human health, natural populations. These three parameters, the assess the concentrations. First is assess the concentration dependent hazard a chemical may present, human health, natural populations. Results typically applied to approval of product use, regulating allowable concentrations in the environment. Some concentration in minute concentration can be used and they are not toxic to our body, can be allowed by the regulatory agency for the benefit of the large populations. Next is type of the studies, in vitro studies, that is a test tube studies, a test tube useful in detecting the potential biochemical and genetic effects. Use model system, bacteria, cultured animals, DNA interactions can be used for the descriptive toxicology. Next is in vivo animals are essential for detecting the health effect. Acute, chronic, multi-generations experimental animals may be treated with a high dose over a lifetime to evaluate the potential to cause cancer. Thus, we can use DMB model and the cell line models for generating the cancer models, and that mimic the human cancer. The completions of all toxicity tests may take 5 or 6 years and is very costly. Molecular and cellular studies in toxicology often supplement the toxicity testing results to help ascertain chemical hazard. They often unravel complex processes that underlie an adverse response. Use of toxicants can help determine the function of proteins in complex network. Now, what private and public sectors invest in toxicity testing that aims to protect the human health? First, particularly the chemical manufacturers, pharmaceutical industries. Our government agencies are keen in developing the toxicological data of marketed products, particularly CSIR, NIPER, ITRC. These are the institutes working for the marketed approval and other toxicological data. Other foreign agencies are US federal agencies, National Toxicological Programs, and EPA. These three agencies are worldwide working for the development of toxicological data.